Welcome to the deep dive. This is where we really try to cut through the noise, get to the core insights from complex information stacks.
Yeah, really dig in.
And today, wow, we are diving into a frontier that honestly feels almost like science fiction. It's all based on the fascinating source material you've shared with us.
Absolutely.
We're looking at how researchers are pushing the boundaries, combining two really powerful cutting-edge approaches to cellular rejuvenation.
That's right. And the sources, they lay out these two fundamental problems with aging cells. First, there's bioenergetic decline. Basically, the cell's powerhouses, the mitochondria, they get old, inefficient.
Right. They start to wear out.
Exactly. And the second thing is epigenetic drift. You can think of that like the cell's instruction manual. It's DNA getting messy. The actual code is fine. But the cell starts, highlighting the wrong bits, crossing stuff out, maybe ignoring important instructions.
Okay, so messy instructions and failing power supplies. And researchers, as the sources point out, they've tackled these separately, right? With some pretty impressive results in the lab.
Oh, definitely. Especially in model organisms. We're talking about work that shows real potential, not just to extend lifespan, but maybe more importantly, health span, how long an organism stays healthy.
Right. But the really big question, the one the source material digs into, is what if you don't just fix one? What if you go after both the energy problem and the messy instruction manual, at the same time?
Concurrently. The hypothesis here, the central idea, is that improving the power supply and resetting the genetic instructions together could create synergistic effects.
Synergistic meaning more than just adding the two effects together, maybe something better.
Potentially, yes, greater than the sum of its parts. That's the hope driving this line of research.
Okay, let's unpack this then, starting with the first approach from the sources. Mitochondrial transfer.
Right. So mitochondrial transfer, sometimes called transplantation. The sources describe it as basically taking healthy, working mitochondria from one source.
Okay.
And delivering them into cells where the mitochondria are old or damaged or just not working well.
And how do they actually get in and work? The sources mention some cool mechanisms.
Yeah, it's pretty neat. They don't just sit there. The recipient cell can actually sort of eat them through processes like endocytosis or macropinocytosis.
So the cell actively takes them in.
Exactly. And once they're inside, they integrate into the cell's own mitochondrial network. They get to work contributing to energy production, that's the OXPHOS, oxidative phosphorylation, and they can help fix faulty signals that the old, damaged mitochondria might have been sending out.
And the sources mentioned this isn't just theoretical, they've seen success in animals.
Yeah, what's really striking is the preclinical success.
Yeah.
We're not just talking subtle cellular changes. The sources detail studies in models of really serious conditions like heart attacks, myocardial infarction, neurodegenerative diseases, metabolic syndrome, showing significant functional improvements. Think about that. Potentially restoring function after a heart attack model or in neurodegeneration just by improving cellular energy.
That's huge.
It is. The sources talk about better bioenergetics, less of those damaging molecules, ROS reactive oxygen species, and importantly, better cell survival. It really could be a big shift.
And is this still just purely lab research, or is it moving towards, actual therapies?
Well, the sources highlight that translational efforts are underway. There are companies, Matrix Bio as mentioned, working on producing clinical-grade mitochondria, figuring out how to make enough of them safely.
Okay.
And they're exploring delivery. How do you get them where they need to go? Intravenous seems possible for systemic delivery, maybe more targeted approaches too. A whole-body transfusion, as the source puts it, might be an aspiration, but getting them delivered systemically is definitely being worked on.
Okay, so that's the energy fix, new mitochondria. What about the other side, the instruction manual, the epigenetics?
Right, so this is where partial epigenetic reprogramming comes in. The idea here is using specific factors. The Yamanaka factors are the famous ones. OCT4, SOX2, KLF4, CMYCAC, mentioned in the source.
The ones that can make stem cells.
Exactly, those are the ones. But here, the key, and the sources really emphasize this, is partial reprogramming.
Partial, meaning not going all the way back.
Precisely. It's about using them carefully, often in cycles, with timed expression. The goal is to rewind the marks of aging, those epigenetic drifts we talked about, to get back to a more youthful gene expression pattern without completely erasing the cell's identity.
Okay. So you don't want your skin cell forgetting it's a skin cell.
Definitely not. Or worse, turning cancerous. That's the risk with full reprogramming. So this partial approach is crucial for safety. It's a delicate balance.
And does this partial approach actually work in living animals?
The sources say yes, based on in vivo mouse studies. These studies are pretty compelling. They show that these partial, often cyclic, reprogramming protocols can lead to real benefits in aged animals.
Like what kind of benefits?
Things like improved muscle regeneration, better function of the pancreas, even enhanced cognitive performance. It suggests you can turn back some aging clocks in tissues without, causing chaos.
How does it actually work at the molecular level, resetting the instructions?
So it induces changes in things called histone modifications. Think of these like little sticky notes on the DNA that tell the cell machinery, read here or ignore this part.
Okay.
Aging messes up these notes. Reprogramming helps fix them. It decreases some marks associated with silencing genes, like H3K9E3, and increases marks associated with active genes, like H3K27C, mentioned in the source. Basically, opening up youthful pathways that got shut down, and maybe closing off ones that shouldn't be active.
So it's cleaning up those highlights and crossouts on the instruction manual.
That's a good way to put it. And interestingly, it also seems to restore communication between the nucleus, the cell's command center, and the mitochondria, the power plants, via specific signaling pathways like AMPK and sirtuins.
Okay, wait. So the reprogramming affects mitochondria too. This sounds like where the two ideas start to connect.
Exactly. The sources point out this fascinating potential link. The cell's metabolic state, how good its energy production is, largely thanks to mitochondria, might actually influence how well the reprogramming works.
So good energy could make the epigenetic reset easier or more effective.
That's the idea, which leads directly to the theoretical basis for combining these two therapies.
Yeah.
The potential synergy we talked about.
Right. It makes intuitive sense, I guess.
It does, doesn't it? If you're doing something super energy intensive, like remodeling the entire way a cell reads its DNA.
Which reprogramming sounds like it is.
Definitely. Having a fresh supply of high energy molecules, ATP, from those newly transferred healthy mitochondria could provide the fuel needed for all that complex work, fixing DNA, changing those histone marks, everything.
Okay, so the mitochondria help the reprogramming. Does it work the other way? Does reprogramming help the mitochondria?
That's the really elegant part of this potential synergy, as laid out in the sources. The epigenetic reset might actually boost the cell's own quality control systems for mitochondria.
How so?
By improving processes like mitophagy. That's the cell's way of clearing out old, damaged mitochondria, and mitochondrial biogenesis, which is building new ones.
I see. So you add good mitochondria, and the reprogramming helps the cell clean house and build more, making it a better environment for those new mitochondria.
Exactly. You're potentially creating a virtuous cycle, a better, more receptive home for the new energy factories.
And the sources mention a specific way researchers are thinking about doing this, something about iPSCs.
Yes, there's this intriguing protocol proposed. The idea is you first take regular body cells, somatic cells, and reprogram them towards induced pluripotent stem cells, iPSCs.
Okay, but you said using iPSCs directly is risky.
Right, so the twist is you don't use the iPSCs themselves, you harvest the mitochondria from them, because these partially reprogrammed or iPSC-derived mitochondria are functionally young.
So you get the young mitochondria without using the potentially risky stem cells directly.
Precisely. Then you transplant those youthful mitochondria into the actual aged cells you want to rejuvenate. It's presented as a clever workaround, leveraging one tech to boost another while trying to sidestep some safety concerns.
And is this actually being tested?
The sources say experimental validation for this specific two-step approach is ongoing. But encouragingly, preliminary data seems to support it.
Okay, so this all sounds incredibly promising, tackling energy and instructions maybe synergistically, but, it sounds almost too good to be true. What are the catches? The sources must mention challenges.
Oh, absolutely. They are very clear about the significant hurdles remaining. This is still very much a frontier.
Like what specifically?
Well, for mitochondrial transfer, the sources list concerns like heteroplasmy. That's where a cell ends up with a mix of its original mitochondrial DNA and the new donated mitochondrial DNA.
Yeah.
That mix could potentially cause problems.
Instead of fixing them.
Right. Then there's immunogenicity. The body's immune system might see the transferred mitochondria as foreign and attack them.
Makes sense.
And also potential mismatches, incompatibilities between the donated mitochondria and the cell's nucleus, especially if they come from genetically different sources.
Okay, what about the reprogramming side, besides the cancer risk we mentioned?
There's still that delicate balance. Ensuring the protocols are perfectly controlled to prevent any oncogenic transformation is paramount. And also making sure the epigenetic changes are precise, hitting only the aging marks you want to hit, without causing unintended off-target effects that mess up normal cell functions.
Yeah, that sounds tricky.
Very. And then overlaying all of this are the regulatory complexities.
Ah, the red tape.
Exactly. The sources make a specific point here. Getting approval for these kinds of advanced combination therapies involving essentially gene and cell therapy components is complex and often slow. Particularly, they note, in places with very strict regulatory frameworks, like many Western jurisdictions.
But they mention it might be different elsewhere.
Yes, the sources contrast this with countries like Japan. Japan has specific legislation, like the Act on the Safety of Regenerative Medicine, which is seen by some as a more, let's say, progressive framework. The implication is that such places might become earlier testing grounds for human trials of these integrated approaches.
Interesting. So what do the sources say is needed to actually overcome all these hurdles? The science, the regulation?
It boils down to a few key things mentioned. More funding, obviously. Much better collaboration between different scientific disciplines. You need the biologists, the engineers, the clinicians, all working together.
Right.
And continued breakthroughs in delivery methods. How do you get these mitochondrial reprogramming factors safely and effectively to the right cells in the body? The sources mention things like nanocarriers or using exosomes. Those little packages cells naturally release as potential delivery vehicles.
Okay, let's try and wrap this deep dive up. The sources you shared, they paint this really compelling vision, don't they? Combining mitochondrial transfer and partial reprogramming. It's not just doing two things, it's presented as a potential integrated strategy.
Yeah, hitting two really fundamental aspects of cellular aging. The energy decline and the messy instruction manual potentially at the same time, maybe with synergistic benefits.
And the goal, the vision presented, is aiming for lasting rejuvenation effects. Not just in a dish, but maybe across different tissues in the body, resetting the power supply and the cell's programming.
That's the ultimate potential discussed.
But, and it's a big but, we have to remember what the sources clearly state. Despite the excitement, there are major scientific and regulatory mountains still to climb. This isn't happening tomorrow.
Absolutely critical to keep that perspective. It's a frontier with huge potential, but also huge challenges.
So thinking about the listener, why is this specific combination strategy, this integrated approach, something worth watching for you?
Well, it addresses those core aging hallmarks in a novel way.
And maybe consider this final thought, building directly on what the sources highlighted about regulations. If regulatory pathways are a major bottleneck, and there's this contrast between, say, Western countries and maybe more progressive frameworks like Japan's, what could that difference in regulatory speed actually mean for how quickly therapies like this truly groundbreaking combination therapies aiming to tackle aging itself might become accessible to you in the future?
Yeah, that's something worth pondering, isn't it?
Yeah.
How science translates and how policy shapes that path.